Hello and welcome. My name is Robert Kowal from the Research Campus Stimulate in Magdeburg, Germany. I would like to present our research on the specific absorption rate in a dedicated birdcage coil for neonatal MRI. When dealing with the diagnostics of neonatal brain injuries, MR imaging is often the most recommended modality due to high soft tissue contrast and relatively safe examinations. Standard systems and adult coils are not optimized for these kind of patients. With the dedicated MR system, of which RF coils are an essential part, one can improve the overall image quality. When we excite the patient, it has to always be ensured the power absorption, which can be quantified as the specific absorption rate, is limited to avoid undesirable tissue heating. This is of even higher importance when we talk about neonates who are still unable to communicate any discomforts. The IEC states SAR limits that must not be exceeded in order to prevent harmful effects. An established procedure is to simulate the electromagnetic fields based on models of the coil and the patient, and thus estimate the maximum acceptable RF input powers for this whole system. Our simulation setup consists of a coil model which was created directly from the PCB production data to preserve details. The birdcage coil's diameter is here approximately 26 cm. As a patient, a heterogeneous voxel model of a baby was used from the Helmholtz-Zentrum in Munich. The placement for the head-centered excitation at 1.5 Tesla B0 field strength is shown in the cross-sectional images to the left. From the simulation, the 10-gram average SAR values were computed and are visualized for 1 watt of input power. Shown are a frontal view as well as the coronal and sagittal cross-sections. Absorption is mainly focused in the effective body volume inside the coil and decreases rapidly inside the patient. A clear hotspot can be seen at the neck region with SAR values above 0.12 watts per kilogram. As a consequence, this leads to localized heating over time, which is also advancing into deeper tissues like the brain. The local SAR maximum was simulated to be about 4 times higher than the average head SAR. With regards to IEC levels, the local SAR would therefore limit the maximum excitation power before other limits are exceeded. In addition to simulation, validation experiments were performed on the basis of ASTM standard 2182 to support the simulation results. A cuboid phantom was placed inside the birdcage coil and the local temperature was measured at 4 positions using fiber optic sensors. The birdcage was excited in quadrature with a continuous sinusoidal signal using a function generator. The signal power was amplified to about 100 watts before being split by the quadrature hybrid. Using 2 directional couplers and a spectrum analyzer, the input powers of the coil could be measured, which is needed to compare with the corresponding simulation. Here on the left image, the center position of the phantom can be seen, with the temperature sensors in blue fixated in the gel. The gel itself is made from a cellulose salt-water mixture, which mimics typical tissue electromagnetically, and its effective mass matches the baby phantom from before. On the right, comparing with a similar simulation, a qualitative agreement can be identified with best matching values at high SAR levels and average deviation of about 30%. This kind of validation gives primarily evidence for the quality of modeling and simulation of the coil. As an outlook, we suggest 3D printing of geometrically realistic human-like phantoms to be compared with their simulated SAR. These would have to still be filled with a homogeneous gel, but first simulations suggest only little deviations for the frequency of interest. These more complex phantoms would allow for further investigation of the simulated hotspot in the neonate model. This I would like to thank you for your attention.